seminar speaker, Dr. Paul Franson. He is one of our own graduates, and I asked him to come back and talk about the research he's been doing since he left. So just by way of introduction, Paul received his bachelor's degree in integrated biology from the Brigham Young University. And from there, he came to Rutgers and he worked on a PhD, and he did receive that. And from here, he went to the Smithsonian, to their data science lab, where he was a research scientist. And he actually continues to be connected with that lab as a research associate. And a couple of years ago, he landed a position, assistant professor position at Brigham Young University. So he's back to his original alma mater. And with that, Paul, I'll turn it over to you.
Great, thank you, Dr. Hamilton. That was perfect. Yeah, so I'm excited just to talk a little bit about some of the stuff we've been doing with insect genomics. This is some work that I kind of started a little bit when I was at Rutgers. Our first inklings of kind of using genomes to understand insects happened as a graduate student. So I'm really excited to be back virtually, at least, and talking with Rutgers. I really appreciated my time as a PhD student there. It was a time of immense growth for me, but also a really good time. I always tell my students that my PhD was my favorite part of my life thus far. I think it was really great. So I'm just going to talk about a couple of different case studies of using insect genomics, trying to understand a little bit about genome evolution. But first, I thought I would just start by sharing some of my motivations for pursuing this type of research. So it's always nice to give a talk in an entomology department because you can appreciate the diversity that you can see in these two museum drawers. So these are two museum drawers that are from the Smithsonian National Museum of Natural History. And I worked there for 3 years right after my PhD, and it was a wonderful time and I loved it. It was kind of a dream come true. And a lot of the time I would spend time looking at museum drawers, and here's two drawers of caddisflies, which is the group of insects that I'm most interested in. And a lot of people, kind of the general public, maybe if I took someone on a tour of the back rooms of the Smithsonian, might look at these and think they're a couple brown moths. But to me, as an entomologist, these museum drawers represent a kind of amazing, almost dizzying array of diversity, both in color and form, in places where the insects were collected, and in what they do. And I'm really motivated by trying to understand the basis of that biodiversity, how it arose, and what genomic processes may have given rise to it. And I've really been lucky during my previous position at the Smithsonian and continuing collaborations to work on all sorts of organisms. So this is just a plate that shows a lot of the organisms on.
Might volunteer at the COVID vaccine clinic for Monmouth.
Sorry. Genomes that I've worked on. So I've worked on genomes from species from mammals to plants and to insects. But after all of that work, insect genomes are still the most interesting to me. And so the primary questions that kind of focus my research are, how is biodiversity generated? So this beautiful array of diversity that we observe, how is that biodiversity maintained? And what role does the genome play in these processes? And then I really want to understand is, can we start to answer these first three questions using comparative genomics methods? And I think insects are the perfect model because they're super diverse. As you all know, I don't have to preach about insects, but the most diverse group of animals on the planet. They inhabit all sorts of different ecological niches, and they're really good models for understanding the generation and maintenance of biodiversity. So I've broken up my talk into kind of three different pieces. The first is to just talk about the state of insect genomics. Where are we? So the first insect genome, the Drosophila melanogaster, was published in the year 2000. And so 20 years from that, where are we in insect genomics? Secondly, a couple more kind of focus studies. First, what role does introgression play in the rapid radiation of heliconius butterflies? And then second, a little bit about Caddisfly genomics, thinking about silk genotype and environment interactions and the role of repetitive elements in genome size expansion. So first, what is the state of insect genomics? So a colleague and I, during COVID times, I was taking most of my meetings in my backyard because my wife and I were swapping with the kids, decided that we would take advantage of some time not in the field and not in the lab to see what's going on with insect genomics. So I downloaded all of the insect genomes from GenBank. And one of the first things that we kind of discovered is that aquatic insect genomes, there were many fewer aquatic insect genome assemblies than you would expect, given their diversity. So aquatic insects make up about 1/10 of all insect species. However, only about 1/20 of, or 1/22 of the genome assemblies available on GenBank are aquatic insects. And not only that, but aquatic insect genomes were of lower quality. So this was our first takeaway, and we published a little paper on that over the summer. But we've been digging into the data a little bit more, along with Scott Hotaling, who's a colleague of mine at Washington State, and one of my undergraduate students in bioinformatics is Ashlyn Powell, to look a little bit more closely at these insect genomes. And one of the first things that you can notice is that these orders in blue are insect orders that have at least one genome assembly representing that order. But those in red down here are orders that lack a genome assembly. And so out of the over 30 insect orders, there's something like 11 that don't even have any genome assembly at all. And there are additional 6 orders that are represented by a single genome assembly. And we also really noticed the power of long-read sequencing technology. So these plots basically show the contig 50, which is a kind of a surrogate for contiguity of a genome. So if you're thinking about a genome assembly as being a puzzle, it's basically how well that puzzle is put together and how many pieces the final puzzle is put together in. And what we found is, which is kind of unsurprising, but hadn't actually really been shown like this before, is that those genomes that were generated using long-read sequencing technologies or bigger puzzle pieces ended up with more complete final assemblies. So the final puzzle was more complete. We also dug in a little bit deeper to look at gene completeness. So there's a piece of software that has determined genes that should be shared across all of insects. So basically every insect genome should have a majority of these genes in their genome. And it's a method that you use to assess how well an assembly works. So if assembly worked really well, you would expect to find, 95 to 98% of those genes in that assembly. And what we see is that genome quality really varies across insects. So this is a plot where the light blue shows complete genes found in the assembly, whereas the red is missing genes. And so as you can see, there are many genomes that are really good. So these types of genomes right here, but there are also a lot of genomes that are actually quite poor in terms of gene completeness. And then we looked at this with regards to long versus short read sequencing again, and found that long read sequencing genomes are more complete than short read sequencing genomes, also less fragmented, and kind of interestingly, end up with recovering more duplications. And so this was exciting, and but kind of what we discovered from this is that the quality of the genomes is so variable that it's really difficult to make any broad scale conclusions based on the current set of genome assemblies from insects. So if we wanted to look at one particular gene family, for example, we'd really have to control for quality since all of them have been generated using different methods. So in conclusion, there's about 600 insect species that have genome assemblies available. There are more than 1,000 genome assemblies on there, but most of them are duplicates. So Drosophila melanogaster, for example, has dozens of genome assemblies. The aquatic insect genomes are underrepresented. Genome quality varies widely, but long read based genome assemblies are definitely superior. But I really think that we need more sequencing in order to ascertain broad scale patterns of genome evolution in insects. And so I'm just going to talk now about a couple of targeted sequencing projects that we've started in order to try to better understand how to use comparative genomics in a fine-grained way to kind of understand a little bit more about genome evolution. So the first is the role of introgression in the rapid radiation of Heliconius butterflies. So this is a project that I started with Jim Mallett's lab at Harvard, and there's a couple of grad students involved, Nate Edelman and Maya Miyagi. And we decided to start a project on Heliconius butterflies. And first, I'll give you a little bit of a background about what the kind of question that we were looking at here. So there's a nice review paper on introgression and population mixing in PLOS Biology, published in 2019. And this plot is, or these figures are from that review paper. So in general, we think about coexistence of different groups of organisms in this way, that there are, individual phenotypes, and those phenotypes correspond with a distinct ancestry. However, as we've sequenced more and more data, we've realized that it's actually a little more complicated. And in some cases, you might think that, oh, okay, they can just mix, and you'll have some sort of mixture of ancestry and of phenotypes. There's cases where some can invade and hybridize, and some of those invasive genotypes will exist in the population, but you may not be able to observe it. It's cryptic. You may not be able to observe it in the phenotypes. There's also hybrid zones or clines where there's varying degrees of hybridization across the geographic gradient. And kind of lastly, or second to last, there's this possibility of introgression. So this is what we're looking at here. And so this is when there's some sort of hybridization event, which are frequently not non-adaptive, but then that hybrid will back cross into the ancestral population, and some genes will be left behind. And those genes are often thought to be adaptive, and people have referred to this as adaptive introgression. So when there's a hybridization event and then the genes are back crossed back into the original population. So as you can see, this fish with a green fin, maybe there's something about a green fin that ends up being adaptive, and they picked it up from the genes from another organism. And lastly, there's a case where new species can arise via hybridization. And so how common is hybridization? Well, it's been studied in several different systems. So the monkey flowers are a good model for those. It's been found in beetles, birds, and even mammals. The snowshoe hare, there's evidence that adaptive introgression is responsible for coats changing colors. And so why should we care about this sort of hybridization? Well, first off, it helps us understand ourselves, right? So we as Homo sapiens are actually, part made of Denisovan and Neanderthal DNA, because there were ancient hybridization events that occurred between populations of Homo sapiens and these two groups of other hominids. And this is important because we're learning more and more that those alleles that we inherited from Neanderthals or from Denisovans can have an effect on our phenotypes. And so this was a paper published last year that showed that there was one major genetic risk factor for severe COVID-19, and that was inherited from Neanderthals. So those people who had that risk factor maintained that particular allele from an ancient hybridization event with Neanderthals. And this is important for lots of folks at Rutgers who are studying mosquitoes. We know that there's been massive hybridization in many important mosquito lineages. And in order to understand how to eradicate those mosquitoes, we really have to understand their evolutionary histories. And then lastly, we know that in the Mountain West, at least, people talk a lot about these hybrid trout that are taking over and invading places where native cutthroat trout, for example, in the Mountain West exist. And so hybridization does affect us in many different ways. But more than that, it's interesting from an evolutionary point of view because it gives us a new idea of how evolution works. And one of the things that has been observed is that hybridization can be common during rapid radiations. So when I talk about rapid radiations, this is when multiple species arise in a short geologic timescale. So one example of this, a classic example, are Darwin's finches, and they arose over the course of 1 million years. So that's a very rapid radiation. There's also tomatoes, which this radiation dates about 2.5 million years. And in both of these cases, they found evidence of hybridization. And so kind of the keys to facilitate hybridization is rapid radiation and geographic overlap. And so similar to those two cases, we know that Heliconius butterflies diversified rapidly. So Heliconius butterflies are distributed primarily in the neotropics. And here's a phylogeny that basically shows Heliconius, the genus Heliconius, in relation with their outgroups. And what the color of these branches indicate is the speciation rates. And so as you can see from the colors, this genus radiated much more quickly than its most close relatives, its closest relatives. And so it has that and in addition to radiating quickly, we do see a lot of geographic overlap of many of these species. And Heliconius has been popular for a long time, and you maybe have read about them in textbooks because they generate these kind of interesting mimicry complexes or mimicry rings. So they're Mullerian mimics, which means that a toxic organism is mimicking another toxic organism to avoid predation. And so Heliconius are toxic, and they mimic each other. And so this is one species of Heliconius, Heliconius melpomene, and it has multiple color morphs. So in Peru, it looks like this. However, in Panama, it looks like that, which is totally different. And you can take another species of Heliconius distributed in the same places, and you'll notice that Heliconius timoretta in Peru more closely resembles Heliconius melpomene in Peru than it does members of its own species in Panama. And so this is a really interesting observation, and it's been observed, or it's been hypothesized, that these color patterns are actually passed via hybridization. And this has been looked at a little bit, not necessarily with whole genome sequences, but with genome reduction methods. And they have found that there is conflicting signals in the phylogeny of these species. And Jim Mallett, whose lab we worked with on this paper, also has a model for how frequently color pattern loci have been transferred via introgression across species boundaries. However, no one had really looked at this with genome-wide data or with whole genome sequencing data. And so we decided that we would test this out and observe how frequent introgression is throughout the Heliconius radiation. And then also what kind of genome-wide effects on Heliconius evolution do those introgression events facilitate. And so we decided to sequence 20 new genomes and assemble de novo throughout the radiation of Heliconius. And so these bolded species are the ones that we selected. And we were able to come up with a project that we thought was really exciting. And I'll just talk about it now. It was published in 2019. And so the first thing that we did is that we came up with the phylogeny of the highest quality assemblies. So we looked genome-wide. We first had to align all the data, which was no small task. We used a non-reference alignment program called Progressive Cactus, which has been used in subsequent papers now, but we were kind of the first ones to use it in a big comparative framework. And something that you'll notice here is that despite using whole genome data, we were still unable to resolve some parts of the tree. So this sylvaniform clade was unresolved as a polytomy. There was just not enough signal, even using the entire genome. What we also noticed is that we then cut the genome into pieces, 10,000 base pair pieces, and made trees for each of those 10,000 base pair chunks. And these lines in the background are those phylogenies from each 10,000 base pair chunk. And what you can observe from this is that there is a lot of discordance between these 10,000 base pair chunks and the species tree that we generated. And so we've been generating species trees. I've been thinking about trees since Darwin, right? So Darwin wrote about these. And even before Darwin, there were philosophers that thought about life as being tree-like. And this is from Darwin's Notebook B, famous drawing where he showed this tree. And you see that it's mostly a bifurcating tree or trifurcating in some cases, where there's an ancestor and that splits into species, but there's no species after these lineages separate. There is no hybridization between species. So that's something that he didn't necessarily think about. And it's just something that we're starting to think about more as we observed whole genome data. And so this is something that we call gene tree species tree conflict. So say that there is a species tree, and let's just assume that this species tree is representative of the true evolutionary history of these three taxa. So A, B, and C, right? But frequently you may get conflicting trees. So this shows the same taxa, but different relationships. So B is more closely related to C here, while A is more closely related to B there. And ideally, the gene trees, which will be represented by this tree within the species tree, should match with the true evolutionary history of those groups, right? That's something that we usually assume, especially in concatenated based phylogenetic methods. However, it's possible that because of ancestral polymorphism and a process that we call incomplete lineage sorting, that you would get a discordant tree just because there were polymorphic alleles in the ancestral population, and those didn't sort along lineages perfectly after the speciation event. And so these become fixed after speciation, even though there's polymorphism in this branch leading to it. And that will give you conflicting signal because B in this case is more closely related to C. The other reason that you might observe this tree is not because of incomplete lineage sorting, but because of a process of introgression, where there's a hybridization event among species and genes are passed from one species into another. And so this would be an example of introgression. And then this signal, whatever signal was here, is erased because of that introgression event. And so we wanted to look at this a little bit more closely in one particular clade where we noticed a lot of discordance. And this was the Eratosera clade in Heliconius. And so first what we did is that instead of using a strictly bifurcating tree phylogenetic tree generation method, we decided to use a network method that allowed for reticulations. And this was the most highly supported network based on that method, where the dark red are very highly supported introgression events, where the dotted lines are moderately supported reticulation events. And so we find that a network is actually more highly supported than a strict tree to represent the evolutionary history of these. But we also wanted to look at this across the entire genome and see how the phylogeny changes across the genome. So we took 50,000 base pair sliding windows and generated trees. And the 8 most common trees that we found for this clade account for 98% of all of the windows. And so these are the 8 most common trees that we discovered, with 2 trees totally dominating the signal that have kind of one difference, in that in this tree, Telecife is sister with Hecalesia, where in this tree, Hecalesia is more closely related to the Eratohymera group. And so together, these represent about 70% of the genome. And you can plot these across chromosomes, right? So we had chromosome information, and this is how the phylogeny changes across these different chromosomes. And one of the things that I like to do when I first get a graph is kind of sit back and let my eyes glaze over and see if there's any patterns I notice. And one of the patterns that we noticed was that longer chromosomes seem to have more blue trees, and the shorter chromosomes seem to have more red trees. And so we investigated this a little bit further and found that recombination rate was correlated with topology. In this case, shorter chromosomes had more red trees and longer chromosomes had more blue trees. And our hypothesis is that since on average in Heliconius, there is 1 crossing over event per meiotic division, that long chromosomes have fewer recombination events per base pair than short chromosomes. And so we generated a genetic map and found that that was true. So these red are the short chromosomes. As you can see, the recombination rate is much higher on the red chromosomes. This is the red tree, the red trees. And the red trees are also more highly much more enriched in those chromosomes with a high recombination rate. And so this is consistent with this idea that that most introgression would be maladaptive. But introgression in spaces with high recombination rates would allow for quicker purging of the deleterious alleles and maintaining the preferable alleles. And so you might imagine that introgress loci would be more common in areas of the chromosome that have a high recombination rate. And that's just what we found here. So the second thing that we want to do is say, okay, well, we found this gene tree, species tree conflict, but how do we know it's not just ancestral polymorphism or incomplete lineage sorting? And so again, like we talked about earlier, here's the true species tree and here's a conflicting tree, and they can arise either via incomplete lineage sorting or via introgression. And there's something that you might notice about the difference between these two scenarios, and that is that the internal branch lengths are variable. So an introgression event has a much longer internal branch length than an incomplete lineage sorting event. And it turns out that you can just come up with a simple mixture model, and you can estimate the probability of each of these events given all possible triplet trees and estimating the internal branch lengths. And so we implemented this into a software called Quibble that allows you to estimate the probability of introgression versus, so this is the introgression distribution, and this is the incomplete lineage sorting distribution. And depending on where your tree falls with internal branch lengths, you can estimate the probability of either scenario. And so using this method, we estimated overall introgression across the entire genome, and we found that of the discordant triplet topologies, 70%, about around 70% arose via introgression rather than incomplete lineage sorting. And this accounted for 20% of all triplet topologies across the entire genome. So 20% of the genomes in these in this Eratosera clade arose via this process of introgression. We also dug in a little bit deeper on a few things, and I'll just share one here. And you see this little box of dark green on chromosome 15. This was a pretty rare topology, so we wanted to investigate it further. And upon doing that, we found that it's due to an inversion on chromosome 15. We identified those breakpoints, and that inversion has an 80% probability of being historically introgressed, and it encompasses a region known to control mimetic color pattern. And so this was evidence that indeed these mimetic color patterns are being passed along to each other via adaptive introgression. And so our conclusions from this is that the heliconius radiation has been characterized by introgression throughout its history, and approximately 20% of those loci have a history of introgression, at least one of which was transferred via an inversion. So now we're going to switch gears a little bit and talk about caddisflies and kind of my favorite part of this. And I'll probably only talk about, since I was late, I'll probably only talk about this silk part portion, but there's a lot of interesting things that we're doing with Caddisfly Genomics, and I'd be happy to talk to anyone about them further if they're interested. So caddisflies are kind of the coolest insects on the planet. These are larval caddisflies. They're kind of best known because they have these modified salivary glands from which they spin silk. And with that silk, they use it as a mortar to create really interesting structures. So from fixed retreats, this entire home lined with silk, complete with a capture net, and then they can filter the water through and eat, feast on this net, to portable tube cases where they walk around on the bottom of the rivers like hermit crabs. And so here are some images of caddisflies and case-making caddisflies. And it turns out that you can put a caddisfly in a bed of gold and it will make bling with its silk. And here are some net spinning caddisflies. And so caddisflies, they're very speciose. They're the most speciose of the strictly aquatic insect orders. So they have over 16,000 named species, and this makes them the second largest freshwater radiation on the planet. This diversification is thought to be directly linked through their use of silk. So to understand caddisflies, you have to understand silk. And this is just a note for the graduate students. I graduated from Rutgers in 2015, and this image is from my dissertation, and I'm about to submit it finally. So sometimes things just take a little while. So when I got to BYU, I had a little more flexibility to work on whatever I wanted. And so I decided that I would look at this idea of caddisfly silk. I didn't know that much about it, but I knew silk was totally vital to understanding caddisflies. And so I linked up with a few colleagues, Xinzhou and Shiki Luo in the Chinese Agricultural University. Xinzhou is actually an alumnus of Rutgers as well. And then Stefan Pauls and Jacqueline Heckenhauer from the Zenkenberg, the Translational Biodiversity Genomics Center, and Russell Stewart from the University of Utah, which is really nice because he's just down the road. What I really wanted to understand is how the silk genotype, this kind of age-old question of how the genotype interacts with its environment to create the phenotype. And so we decided that we'd start to just generate data from each step of the central dogma of molecular biology, right? So we know that DNA is transcribed in RNA, which is translated in protein. So we started to generate data from each of these stages to see if we could link them up and better understand how the genotype relates to the phenotype. And so first, sequencing. This was actually non-trivial. So people have been thinking about caddisfly silk before me. In 2009, Yonamura et al. published the very first partial sequences of the heavy fibrin gene, which is the primary component of caddisfly silk. And what they found is that there were these repeat motifs that were repeated over and over again. But what they also found is they could not sequence through the entire gene. So they were only able to sequence the N terminus and then the first couple repeats, and then the C terminus and the last few repeats. Similarly, my colleague Russell Stewart and colleagues generated a transcriptome and found the same thing. Very similar repeats, but again, we're unable to sequence through the entire gene sequence. And there's kind of a simple reason for this. The heavy fibrin chain gene is really long. So imagine this blue is the N terminus and the end of the blue is the C terminus. And it's made of these repeat motifs that make up the entire internal sequence of the gene. And so they were using short read sequencing technologies. And if those short reads, so just a little 150 base pair read of the transcriptome do not span a repeat motif, then it's impossible to read into all the way through that particular gene. However, we're lucky that long read sequencing technologies are taking off, and they can span an entire repeat region or span the entire gene even. And so our first step was to generate the first high quality Tricopter genome. And so Xin and Shiki and I were in Beijing, and we went out and collected this thing, which is Stenocycle tianmushinensis. And it's important for the Chinese because it's one of the first caddisflies to be described by a Chinese taxonomist. And when I give talks at like an evolution or EEB departments, not everyone agrees with me, but hopefully you all agree with me that this is like an entomologist heaven, right? So this is outside of Beijing, amazingly, at a river, and we collected just a ton of Stenocycle. This is Xin collecting these insects. And we were really excited because finally we were able to read through the entire heavy fibrin chain gene. And it turns out that it was almost it's over 20,000 base pairs long. So it's a long gene. And previous attempts on similar species had only gotten about this far from each end, right? And so with this in hand, we were able to kind of construct an unprecedented view of the density of particular repeat motifs. So each of these blocks represents a different repeat motif. And this is from the N terminus all the way to the C terminus, how they are situated. And then I was at BYU, so I decided I wanted to try to start to sequence some stuff in my own hometown. So I went out to the local Provo River, which contains this guy right here, his Parapsyche elsis. It's a net spinner. We wanted to understand whether the net spinners looked similar to other species that we had worked on. And so we used a kind of an emerging technology called Oxford Nanopore. And it's easy enough that I was able to train undergraduate students to work on it. And this is a histogram of Reedling. So what you can observe here is that the median is higher than the length of that silk gene. So we had a high idea that we were optimistic that we could resolve the silk gene using this technology. And indeed we did. And so we published this in 2019. And here is the silk gene and the repetitive motifs. And so you can see 1A and how it shows up in the rest of the silk gene. 3B is a very common repeat motif, this dark blue, right? But we wanted to go a little bit further. Great, now we can get the silk gene itself, but what can we learn about actually how this silk is interacting with the environment to generate these really marvelous extended phenotypes? And so my colleague just down the road here, Russell Stewart, had done some amazing work on a case-making caddisfly, Hesperophylax magnus. And he found that the silk was heavily phosphorylated. So this is a SEM image. We're using electrodispersive spectrometry to estimate elemental analysis. And he's able to find that the silk fibers, which are these right here, were heavily phosphorylated. He also did some careful measurements to figure out the properties of silk. And so he found that it was viscoelastic. This means that you can stretch it out and it takes about 2 hours to regain its form. And that you can do this as many times as you want, and it can continue to regain its form over and over and over again. They also estimated the toughness and modulus and found that it's an even better material than the human meniscus. And so it's a really cool biomaterial. Lastly, what he found is that if you wash the silk and then input a solution with different concentrations of metal ions, the properties of the silk change. And so they hypothesized that this is because these beta sheets made from these phosphorylated amino acids complexed with metal ions in different ways, making a cross-link protein. And so when immersed in zinc, for example, it was much stronger than if it was cross-linking with magnesium. And so that the metal ions being integrated in the silk have a direct correlation with the strength of that silk. And so we wanted to see if this kind of held true for parapsyche. So we collected a bunch of nets and did some SEM. We got some beautiful images, kind of some of the first images of looking at how the silk is laid down within these nets. This is just 50 microns. We also did some elemental analysis. And this is a 2D map showing the amount of phosphorus across a silk junction. And as you can see, silk from parapsyche is also heavily phosphorylated. But knowing that it's phosphorylated isn't quite enough, because we actually want to know what parts of those peptides contain the phosphorus. So to understand that, we used Tandem mass spec, where you take a protein, digest it into peptides, and you can use mass spec to figure out what that peptide sequence is and to pick up which peptides may be phosphorylated. And we picked up these particular repeat motifs and found that these serines were frequently phosphorylated. And so if you look back at our original gene map, these residues that are highlighted in red were the ones that were found to be phosphorylated using this tandem mass spec technique. We also found that the silk integrated metal ions from, this is calcium. So you can see calcium is also integrated in the silk. And you can look at this another way by just doing a line scan across a silk fiber and see that it's enriched for calcium and even magnesium. And lastly, we use transmission electron microscopy to evaluate the size of the silk gland precursors. So we wanted to look at Hesperophylax, a case maker, versus a fixed retreat maker, and see if the silk precursors differed. And we converged on these same kind of silk precursor shapes. However, the size was divergent between the net spinners and the case makers. But kind of the coolest thing that we found is that we also did elemental analysis on the silk itself with mass spec and the silk gland precursors. And we found that in the net itself, the metal to phosphorus molar ratio was almost 1-to-1. So a lot of calcium integrated into that net silk. However, if you look at the silk gland precursors, silk precursors, there's almost no metal in there. So if you can notice the difference in the y-axis here. And so from this, we determined that when the silk is exiting the labial glands of these caddisflies, it does not have any metals. Rather, the silk, as it's hardening, gathers metals from the aqueous environment. And the distribution of metals in the aqueous environment has a direct effect on the phenotypes, as Russell found earlier. And so in conclusion, we found that long-read sequencing technologies can resolve long, heavily repetitive silk genes, and that the caddisfly silk phenotype is dependent on the type of metal ions cross-linking with those phosphorylated serines, and larval caddisflies are picking these metal ions up from their environment. So caddisflies have long been used as biomonitors, and so this maybe gives some indication of why they're differentially sensitive to different levels of metal pollution. And I'm going to have to skip this last section, but there's been some interesting new work on genome size evolution in caddisflies. We just published a paper 2 days ago. So if you want to look at that, it was led by a great undergraduate student in my lab. And we found that caddisfly genome size is really driven by repetitive element activity. And now we're looking across a whole bunch of caddisfly genomes, 25 of them, and finding kind of the same patterns. But with that, I'll just end with some future directions. I want to spend my career looking at genome evolution in insects and figuring out if there's patterns. For example, I'm really interested in aquatic insects. Aquatic insects, insects have invaded freshwater over 50 times. So is there something about insect genomes that make them better able to invade freshwater? I'm also really interested in silk and caddisflies and kind of have this nascent interest in repetitive element activity. I never thought I'd be interested in repeats until I generated a bunch of insect genomes where it seems that repeats are really interesting. And so with that, I'd like to acknowledge all of the people who have helped me in all of this. There's lots of folks at BYU who have helped, some graduate students, whole host of undergraduate students and external collaborators, and I'd like to thank you for listening.
Great, thank you, Paul, for the excellent presentation. Everybody can ask questions now. If you like, please turn on video so that everybody can see you.
So Paul, I've got a question for you.
Yeah.
It has nothing to do really with all that science you just talked about, but in your insect cases of the caddisflies, I'm always amazed at the diversity of color patterns in those, because what I'm familiar with here in the East, none of them are as spectacular as all of those. Is there a part of the world where that color patterning is better or more is increased as opposed to others?
Yeah, and so are you talking about the color patterns on the wings or the larval cases?
Oh, well, I could go there too. I was talking about the adults.
Yeah, the adults. Okay, yeah. So the adults, it's much more common to find colorful caddisflies in the tropics.
Okay.
And we don't know exactly why, but you might have also noticed that the more colorful they are, the bigger their eyes are. And so there might be something with mimicry or with sexual selection. And that's another thing we'd really want to understand. And there's a graduate student in Nipam Patel's lab, at Evo Devo lab, who's looking at the evolution of scales in caddisflies. So there are some caddisflies that have scales. Most of them have hairs, but some of these colorful ones have scales. And so hopefully we'll have more information on how that works soon. But yeah, they are more common in the tropics.
Okay.
Yeah, I'll ask a question. Hey man, how you doing?
Good. How are you?
It was an awesome talk.
I have another question, but Dana got there first.
All right.
Sure. Find it real quick, actually. All the way back to the base of the heliconius analysis. You guys said you were using F1s?
No, so most of them were wild caught, but we sequenced one F1.
Okay.
Yeah, so that was probably a little misleading on there. So yeah, most of them were wild, but then we had an F1 hybrid in there.
Okay. So
and we didn't use that in any of the tree-based analyses.
If most of the data was from wild types, and you're using, I guess, a haplotype assembly, right? So everything was assembled and you have a consensus. Do you have any idea how much variation there actually is if you were to take like the minority consensus, if you had some short read sequencing or something like that, and tried to actually figure out which alleles were actually represented in your assembly versus those that weren't?
Yeah, that's an interesting idea. So these were all short read genomes, and we used this method Discovart de novo, which actually does phase them, but we couldn't find any tools that worked with phased graph genomes, and so we did collapse them all. But I think that would be really interesting, and because the story in some cases really could change.
Yeah, that's what I'm wondering, because we had, even with just protists that don't have sex, there's really a discordance in some cases between what you get with the majority consensus assembly, even with the phase assembly, versus the unzipped haplotype. So cool.
Yeah, no, that would be a really interesting thing to look at, because and we did estimate things like, we estimated things like heterozygosity, and they're very heterozygous. All of these wild caught things. So it's very possible that there could be a different signal hidden in the other genome. So
yeah, I like Silkomics.
Yeah.
Did you find that?
That was actually not mine. That was my University of Utah colleague who came up with that. I'm a little anti everything being omics, but I let it slide this time, because we really did do silkomics. We had transcriptomics, genomics, and proteomics, so I figured it was okay.
I had a question, but I'm thinking of how I can rephrase it, so I'll let someone go. I really like your. Yeah, sorry. I may have cut you off then. But I am curious, like this kind of whole field of genome size evolution. We had a talk last semester, and I asked him if you had crossed paths. His name is Carl Gelman. He works with Spencer Johnson at Texas A&M. He's a postdoc now, but he did like his whole thing was genome size in Drosophila. So I see like a lot of overlap, but I guess you guys haven't crossed paths.
Yeah, no, I'm kind of new to this. I mean, the only reason we got interested in genome size is just because we said, oh, caddisflies have a lot of genome size variation. So I've read some of the stuff from Johnston's lab. But yeah, I don't know the community all that well, other than just papers that I've read.
Yeah, I know this was in Drosophila, and then he got into kind of forensic species and looking at genome size. But it's pretty interesting when we do the comparisons and we see kind of big discrepancies in like what genome size there are. I know one of the graduate students is working on a genome assembly now, and he's found that the species have different like size genomes, right, Grayson? I don't know if that was going to be your question.
No, but that's the gist of it. So yeah, I guess I know Dr. Gelman too. So I guess he's one thing that's, I guess, interesting about his research is he also trying to look at how genome size, maybe tied to life history traits. But I don't know if he's found anything just yet.
Yeah, so we're really interested in that same question. So especially in plants early on, they found that genome size evolution, and especially repetitive element activity, was correlated with particular life history traits. And we find that in caddisflies too. So the case makers inhabit a much wider variety of habitats than fixed retreat makers that have smaller genomes and less repetitive element activity. And there's these ideas, and these are all just ideas. So don't think that I'm saying that this is what's happening, because we haven't looked at it closely enough to know. So I'm waving my hands right now. But there appears to be an interesting correlation of repetitive element activity and the variety of habitats that an organism inhabits. And there's this idea of repetitive element activity being able to pick up genes and move them around the genome and potentially increase variation, which then leads to neofunctionalization of particular protein coding genes. Because just like introgression allows for, the reason that, most eukaryotes have sex is because of variation, right? And so why not expand that a little bit with introgression? So that's why introgression can be adaptive, because it's new variation being introduced into your genome. But similarly, repetitive element activity, we actually do have evidence that there are repetitive elements that are inserting into genes and then moving those genes all throughout the genome in these caddisflies. We need more experimental evidence to figure out if those genes are actually functional or not. But it is kind of make a nice story of how the genome might kind of, move around in order to allow more variation for natural selection to act upon. So that's I'm writing a grant proposal right now just to look at that within caddisflies, because I think it's really interesting. But yeah, I guess it's all hand wavy right now.
I guess that kind of feeds into a question I had. So are these introgression events usually tied to something that would be like an adaptive phenotype? So you have all these mimicking butterflies that have sort of developed similar coloration, but is that seen in other groups?
Yeah, so it's been observed in other groups. And one of the problems is that, there was a lot of stuff that was introgressed in those, right? And so we don't know necessarily what all those regions of the genome do, but most of what's been done with adaptive introgression, a lot of it has to do with color patterns. So in the snowshoe hares, for example, GUDI, which is the gene that governs a lot of color patterning in mammals, has been adaptively introgressed. And then in Heliconius, we find cortex and optics, which are both color pattern loci, which have also been transferred as a result of introgression. And so this is kind of a tractable, it's a very easy phenotype to observe. And then we actually have pretty good connections of the genes with those phenotypes. And so I think that's why that particular trait has been studied quite a bit, but it'd be really interesting to look at other traits that could be adaptive as well. But yeah, those are the main things that we know of, just because we have a lot of work to do to find out what the other genes are doing.
So in regards to kind of like the color adaptation, so you have these kind of morphological ways that they are different. Have you ever thought of, or has there been work done, and I have no idea, done on kind of social insect genome size? Because we know that the proteins undergo sub-functionalization between different castes based on their roles. But then I guess you have this kind of behavioral approach. And I wonder if that the size of the genome has some kind of effect on how this is determined in social insects too, if that makes sense.
Yeah, I think so. Let me just share this. So this kind of little dive that we did into the state of insect genomics. One of these panels is the assembly sizes. So I'm trying to see. So here are bees. Formicity. Doesn't look like there's a ton of genome size evolution. What else do we want to look at?
I worked on like
Termites, maybe.
For my PhD, I worked on fire ants, and kind of just as I was just about to defend, they like redid the assembly because it was like really bad the first time they did it. It was like a combination of like shotgun sequencing and all different techniques kind of meshed into one thing, and then they just kind of redid the assembly.
Yeah.
But I guess I mean, social insects are cool, so I wonder how much it like plays into like the behavioral traits as well as morphological traits.
Yeah, that'd be interesting to look at. I don't know the answer to that question, but
I don't know that anyone has, because I think I asked Carl Jellman the same question, and I don't think, maybe it's just I'm interested in that. But I loved your talk. It's great to see kind of more genetics, and it really makes us consider kind of when we do things like transcriptomics and protein expression, it really makes you think like where that really originates. So
yeah, great. Yeah, thanks.
Any other questions? If not, we will end because it's already 12:03. Thank you, Paul, for the excellent seminar.
I'm sorry again for my late arrival,
No problem.
but I'm really grateful to come. So we'll have to do it again sometime when COVID's over, so I can come back and visit you guys.
That'd be great.
Yeah, we'll have a 5 minutes break, and then the students can remain here for further discussion.
Okay, so if I just
Yeah, just
turn off my,
stay here, right?
come back for 5 minutes.
Yes.
Yeah.
I'll just turn off my video for a second and
Yes.
go take a break. Yep,
Okay.
thanks.
No problem.
Thanks, everybody.
Thanks.
So Chloe,
yeah,
did you or Cara send out questions for today? Or
so the questions that I sent you, they're the group 2 questions.
okay,
So we did group 1 last week, and then it'll
Right.
be group 2
Okay.
this week.
just checking.
Yeah, and then we'll see. I kind of it's at 2 different times, but I think you might miss the second one.
Well, if your experience with having a session for the async folks for questions and nobody shows up, I'll be
Yeah.
right on time.
I think if Tim Lampasona is here, he can kind of validate that too. Me and Tim would do open office hours, and me and Tim would just chat for an hour.
Okay.
Maybe one student will come, but it's mostly out of like, they kind of want to see what we're up to rather than they have like a question.
Okay. Okay.
But it's good. So I think I told you we started planning a symposium for ESA. Well, I have like 2 symposiums for ESA.
Mhm.
And we kind of have one that's focused on, I don't know if anyone's here. I don't want to like disrupt the session. We have one focused on like kind of like teaching strategies in entomology, like during the pandemic.
Mhm.
And I think like most of the speakers have said that they're coming, but we have one guy that I thought might be like super relevant to like taxonomy based stuff if we can't do that again, because he's kind of figuring out ways that you can do like collections and bringing insects into the classroom when there is no classroom to bring insects, which I thought was interesting. Because I haven't heard of anyone doing that, and I was telling him like, yeah, that was like a huge problem for like insect biology and stuff.
I seem to remember Dana telling me something about somebody that was doing that.
Oh,
You might ask him because he was trying to figure that out for taxonomy last fall.
it's the same person. I don't know if
Oh,
Dana's
okay.
here. It must be the same person. I mean, he told me he was trying to put together like a seminar or workshop and like how to use insects in a classroom when you don't have access to collections and stuff like that. And it wasn't something I thought that anyone would talk about in the seminar, but I was like, that's interesting and kind of goes beyond like my expectations. So
Right. Yeah.
yeah.
Now what do I do?
Right. Yeah. Because I think everyone's kind of in the same boat, like what's going on. But it looks like the insects in the environment syllabus is passing through with no
good.
major discussion points.
We talked about that as a new class that was coming through when I had my meeting with the deans on Wednesday.
Okay. Okay. Well, it was Laura Lawson's idea.
Oh, was it? Well, she wasn't
Yeah.
that.
Okay.
She wasn't that. Well, yeah, she was there because we had to wait for her to get there.
Okay.
Yeah, but they had lots of discussions with me about why don't we have another core certified class. Because
Yeah.
they're just like, well, if you could do that with World of Insects, can't you do it with another one? And I kind of said, well, maybe insects in the environment might be something.
Yeah.
But
That
we also got Tom got in his brain that this course that we haven't taught since 1990 something. It's called Insects in Man.
Okay.
It's on the books.
Yeah, I see it there, and I looked at it and I was like, maybe this could be a cool class. Maybe I'll do something.
Yeah, but
with it.
that name is the name that World of Insects used to have.
Ah.
As
Okay.
the course. I think it's got the same course number. So that's going to be a problem. Yeah.
Ah.
All right. So it looks like Paul is back. I don't see Cheng Lu. Are you there, Cheng Lu?
Sorry, I can't please come back.
No problem. Okay. So again, that was excellent. And so what we do now, Paul, is we've got, when you were here, we only had the graduate student version of seminar class. But now that we have an entomology major, we also have a version for the undergraduates as well.
Oh,
and
great.
so we meet together with the speaker after, and usually over pizza. Of course, we can't do that because of COVID this year. So I'm just going to go through and I'm going to ask each of the students to introduce themselves. And then if they've got a question, ask you a question based on what they read, which they got an opportunity to look at the 3 things you sent me, or based on the your seminar today. And so we'll just start with Patrick.